Okay, good morning, everyone. I'm going to just talk where we left off. Remember that transcription solves a dilemma. That dilemma is getting the information that's in the nucleus, the gene, out into the cytoplasm so that it can be turned into a protein. And we send a messenger molecule here in red called mRNA. It's a little RNA complementary version of a gene. And it's this little guitar pick is called Rna Polymerase. It's the enzyme that reads the DNA and makes the mRNA. Okay. Now, so if you were, the RNA polymerase, what you would do is you'd read an A. You'd read a T, and you would make a little A in your mRNA. Okay. If you read an A, you would put a U there, and if you read a C, you put a G, right? And of course, these bases here, these nitrogenous bases in yellow, are RNA. They're not DNA molecules or DNA nucleotides, which is why there's a U and not a T. Okay, CCC would be GGG, ATT would be UAA. Okay, and then this little molecule would leave the nucleus, float out into the cytoplasm, where it's going to be translated into a protein. All right, and here's another example. Here's your gene. All right, here's your mRNA. So you're going to do practice with that. Now, in your cell, there are three sequences: ACT, ATT, and ATC on a gene. At the end of a gene, right, as you'll notice here, that's ATT, that tell you it's done. All right. Once the little enzyme reads that, it's done, and so it doesn't make an infinitely long or super long mRNA. So that's how it knows how to stop. Okay. Another picture of the same process. There's the little RNA polymerase. It's reading the DNA in blue, and it's making the mRNA here. All right, here's another example of the mRNA, and unfortunately, we're not going to do this big project we're going to do in class because it's just too involved without direct instruction. But all right, so you're going to do some practice. I have a little project where you practice with mutations and stuff, doing some transcription and translation, but right now we're just going to move on to translation. Step 2, we've solved the problem of moving the information from genes into the cytoplasm where they have to be converted into proteins. And here's, in this little picture here, the pink is the mRNA. It moves out into the cytoplasm, right? Here it is. It finds a ribosome, which looks like this weird cartoon hamburger thing. It's got two parts to it, the top bun and the bottom bun. And the mRNA will get sort of read, like a message or like a book, let's say, or like blueprints. And I'll show you how. And the protein, these little orange amino acids are the protein, will get made. Okay, and you're going to learn how to read that and do translation. So the goal of translation is to read the mRNA and convert it into the protein. Okay, it's like reading blueprints or reading a recipe and making the actual meal or the whatever the thing codes for. Okay, mRNA is read in groups of 3 letters. That's why I showed you, if we go back for 1 second, that's why I keep grouping them in 3 letters. See, everything's grouped in 3 letters. I don't know if you noticed that, but groups of 3 letters are like a word, a chemical word, called a codon. Each codon codes for 1 amino acid. Okay? So there's actually 64 combinations of A's, U's, C's, and G's in groups of 3 letters, and 61 of those code for amino acids. All right? And the other 3 are code for stop signal. Okay, the codon actually tells tRNA, a molecule that we're going to talk about in a second, which amino acid to get in the cytoplasm. Okay, and I'm going to show you an animation of this so it makes a lot more sense. I'm just going through like the basic steps and then I'm going to walk through this. All right, so here's a little cartoon version of our ribosome. Remember, it's got the top of the hamburger bun and the bottom of the hamburger bun, the little purple pill-shaped things here is our mRNA in this little drawing. And the reason they're showing you like individual segments is because each one would be a codon. And then there's this tRNA molecule which can read a codon. Right, there's 61 different tRNA molecules, and they can read the codon and they bring over the correct amino acid. All right. tRNA carries the right amino acid to the ribosome. It gets linked together in a dehydration synthesis reaction, and you slowly add a big long chain of amino acids together until you have the protein, at which point the chain of amino acids folds, okay, into a three-dimensional shape. And remember, the shape of a protein gives us its job. So, you know, collagen is a rope-like protein. It helps to hold skin and other tissues together. Keratin makes these kind of real hard bonded proteins together. It makes hair and nails. Lysozyme is an enzyme that's shaped specifically to break the cell wall of bacteria, so it helps us not get infections. Okay, so we're going to talk about the process of translation, and in the next video I'll show you how to translate, or maybe I'll do it in this one. But so let's just walk through the process for a second. I like this animation. Okay, actually we'll do both. It goes through transcription and translation. So here's a cell, a real simple cell. There's the nucleus. Here's the cytoplasm, and here's the cell membrane. A message gets sent to the nucleus to start making a certain protein, and the example we've been using is, because it's springtime now, melanin, right? Sunlight hits your skin cell. This is a skin cell. The DNA gets the message, oh, let's make some more melanin so that we protect this skin cell from DNA damage. Okay, so maybe that would be the example. Inside the nucleus, the DNA opens up for transcription. RNA polymerase gets to do its job. All right. So they're not showing you RNA polymerase, but you know it would be here reading these letters, right? It would move along. It would read a C and add a G. It would read a C and it would make a G. It would read a G. It would make a C. Blah 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 blah. When it reads an A, it adds a U. And this red molecule here, once it's done, is our messenger RNA. Here's a little hole inside of this is the nucleus. It's called a nuclear pore, and that is a spot in which the mRNA can exit. All right, and I know we talked about more details, but that's a basic animation for the transcription. Pardon me. So the mRNA then travels to a ribosome. It's fed through that ribosome. Remember, here's our hamburger. And as it's fed through, groups of 3 letters called codons will be read, and those 3 letters determine which amino acid. All right, in other words, which order of amino acids will be put together, and the order determines how they fold, and how they fold determines the protein's function. Okay, so it all comes down, if you trace it back, it all comes down to the DNA sequence that makes the mRNA sequence, and the mRNA sequence determines which amino acids will be added in which order. So here, all these little funky shapes are different amino acids, ones you know a little bit of. Like, remember you drew way back when you drew glycine, alanine, and serine, and there's 17 more. And so whatever this one is, this little triangle one, they're trying to show you this one will be is coded for by this codon. So this codon, GGC, determines that this triangle amino acid will be added to the chain. How? Because there's a molecule that knows which codon codes for which amino acid, and that molecule does the translating. It's called tRNA, for transfer RNA. Okay, they float around. There's 61 different combinations, and they have on one end something called the anticodon. The anticodon is the complementary code to a codon. Okay, so for example, if the codon on the mRNA says GGC, this one can bind to it because G and C are complementary. Next spot, G and C are complementary, and the next spot, C and G are complementary. Okay, so it'll attach. So, for example, see, GGC can attach to the tRNA molecule that has CCG. Only that one will attach to it. And it can come on in, attach, and it has only one amino acid attached to it. See, it has this binding site that specifically fits, you know, a square amino acid or something, or whatever it is. And it'll bring it in and add it onto the growing chain. Okay, so look, I think it showed whatever. It shows this one's going to attach. Okay, so this specific codon can only attach to this specific anticodon and will drop off this amino acid. Okay, so this codes for this codon, codes for this amino acid, whatever we call it. The first one's always methionine, but at least when we start. Okay, the next codon codes for a different tRNA. If this CCG anticodon tried to attach to this one, it would just bounce off. Okay, so because it doesn't, C and A don't pair. So it has to be the right combination, and then they will do dehydration synthesis and attach to one another. Covalent bond. And it will continue. It will continue until the entire mRNA group of codons is read. We have a chain of amino acids. They fold into a protein, and this weird squiggle thing is the protein. And, you know, that squiggle thing is brown because we're talking about melanin as an example. All right, so this would be the melanin protein, and it would be made in mass, dumped into your, you know, skin cells in the melanocytes, and you would become tanner. Okay, in our example. And you could do this, you know, multiple times per mRNA to make it efficient. And then your cell just breaks it down through dehydration, through hydrolysis, and you could actually reuse these nucleotides. You can make a new mRNA if you need to. You don't just get rid of it. All right. Okay, so that's the basics of translation. What we have to work on next, and maybe I'll turn it into a separate video. I'll decide in real time in a second here. We have to practice translating your maybe we'll try it right now. I'll see if I can squeeze it in. I think 15 minutes. I looked it up. 15 minutes it might be the limit to how big I can make a video. You're going to use this thing. It's called a codon chart. And the codon chart looks it looks intimidating, but it's not that bad. Here's how it works. The tRNA is basically doing this job of reading the 3 letters and deciding which amino acid to add, okay? So the first one, we're reading this codon, AUG, all right? And the codon chart allows you to read the codon to determine which amino acid to add. The A is first. Go to the quadrant with the big A, okay? The U is second. Then within this quadrant, find in the second layer of letters U. There it is. And then the third letter is G. Within this little U slice, find the G. G. So AUG codes for methionine. Now, you're not going to write the whole methionine here. It would be annoying. So just write the first 3 letters. So TAC on the gene makes AUG as a codon, which makes methionine, codes for methionine to be added as the first amino acid. So you're going to write MET dash connecting to, right? That dash means covalently bonded to, whatever this one codes for. AGC, let's find it. That codon codes for AGC, serine. So now we have MET first, dash SER second. All right, GUU. GUU codes for valine. So we have, oops, methionine, serine, valine. So you can use this codon chart. All right, you can either Google it or you can use it from this PowerPoint or whatever, and it's not that bad, but you can use this when you do the hemoglobin mutation lab in a second. So, look, I just showed you an example, all right? So you're going to go through the steps, and CAC codes for CAC codes for histidine, GUG codes for GUG codes for valine, and that's exactly what tRNA will do. I'm going to stop the video because we're almost at 15 minutes, and I'll continue next time. Bye.